As you know, after collection of evidentiary material from crime scene, the same are sent to forensic science laboratory for DNA analysis. The first step in DNA analysis is extraction of DNA from various types of evidentiary materials. For that, there are various methods of DNA extraction and quantification of DNA, because for further processing of the DNA sample after extraction, quantification is must, as you would understand later on. DNA can be extracted from any type of biological sample, for example, blood, semen, saliva and their stains on any surface, organs and tissue including kidney, spleen, gonads, etc., bone and teeth, hair root, hair shaft. For mitochondrial DNA, nail clippings, urine, fecal matter, sweat, tear, earwax, fingerprint which contain touch DNA, skin and muscle tissue, dandruff and sample of plant origin, microorganism, vaginal cell on outer surface of the condom, tobacco spit, cigarette or BD stub and chewing gum encountered in criminal cases. On receipt of such samples in the forensic science laboratory, the first step is to test them for the presence of blood, semen or saliva, etc., and to take a small portion of exhibit for extraction of DNA from it. Extraction procedures may vary according to the type and amount of biological evidence materials. The method chosen should yield sufficient quantity of good quality and high purity DNA. DNA of insufficient quantity and poor quality or degraded DNA may result in a partial DNA profile or failure to give any profile. Low purity DNA may cause interference during subsequent DNA testing. For example, DNA polymerase inhibitors interfere with DNA amplification. Some other consideration for selecting proper DNA extraction methods include adaptability to automation, throughput, simplicity, risk of contamination and cost effectiveness. Now, I shall talk about general principle of DNA extraction. Most of the DNA extraction procedures include the following components and consideration: cell and tissue disruption. Enzyme like proteinase K is used for cell and tissue disruption. However, tissue can also be disrupted by boiling and alkali treatment. Bone and teeth are frozen in liquid nitrogen and then ground to a fine powder. Then next step is lysis of membranes and organelles in the cell. DNA is released from nuclei or mitochondria after lysis of membranes, for which detergents like sodium dodecyl sulfate and sarcosyl for destroying membrane denature and disassociate protein from DNA are used. Buffer systems such as Tris HCl is used to maintain the pH range for avoiding action of degrading enzyme. For disassociating nuclear protein like histones from DNA, high salt concentration is required. to inhibit oxidation process which can damage the DNA, reducing agent like mercaptoethanol or dithiotritol, in short DTT, are used. Endogenous DNases in presence of divalent metal ions catalyze the hydrolysis of DNA. For capturing these metal ions, EDTA or Chilex are used. After that, removal of protein and cytoplasmic contents is carried out. As you know, the constituent of cytoplasm like protein interfere with DNA isolation, which are disassociated from DNA by action of detergent and high salt concentration in lysis buffer. These dissolved proteins are then removed by one or more round of extraction with phenol, phenol chloroform or chloroform isoamyl alcohol mixtures, followed by centrifugation. Alternatively, reversible binding of DNA to a solid matrix like silica gel, which selectively binds DNA, is also used to remove protein and cytoplasmic contaminants. As regards contamination, the target DNA isolated from exhibits is often get contaminated with exogenous DNA while handling and processing the sample. It can happen between person and the sample, between samples, between sample and amplified DNA, or from other organisms. To avoid contamination, the evidence samples are extracted and processed in separate areas or at different times from DNA amplification areas. Evidence and reference sample are processed separately in different areas to avoid contamination between samples. DNA free test tube and aerosol resistant pipette are used and level of contamination is monitored using reagent blanks. Storage of DNA solution. The extracted DNA is stored in TE buffer at 4 degree or 20 degree for short term and at minus 80 degree for long period. Frequent freezing and thawing is avoided to save the DNA strands from breaking. Now, we come to method of extraction. There are several methods of the extraction. First is phenol chloroform organic extraction method. In this method, 3 steps: cell and protein digestion, extraction with organic solvents and concentrating DNA are carried out. Protein digestion is carried out using proteinase K, and then the proteins are removed using mixture of phenol chloroform isoamyl alcohol in the ratio of 25:24:1. Phenol extracts the protein from the aqueous solution. Chloroform being of higher density, the phenol chloroform mixture forms the lower organic phase containing lipids, which is easily separated from the upper aqueous phase containing DNA, while the proteins are located at the interface between the two phases. Isoamyl alcohol is used to reduce foam formation. For concentrating, the DNA is precipitated by adding salts and ethanol. Ethanol exposes the negatively charged phosphate groups of DNA. Presence of cations of ammonium, lithium and sodium in sufficient quantity to neutralize the charge on exposed phosphate residues helps in ethanol precipitation of DNA. Alternatively, ultrafiltration of the aqueous phase using devices like microcone and centricon can be carried out for concentrating DNA. Phenol chloroform extraction yields large size double stranded DNA which can be used for RFLP as well as PCR based analysis. Another method is salting out method for DNA extraction. To circumvent the cumbersome method of deproteinizing cell digest with hazardous organic solvent like phenol and chloroform, Miller et al. In 1988 published a rapid, safe and inexpensive method to simplify the deproteinization procedure. This method involves salting out of cellular proteins by dehydration and precipitation with saturated NaCl solution. In this method, buffy coats of nucleated cells taken in anticoagulant EDTA or ACD solution are suspended in nuclei lysis buffer and the cell lysate are digested overnight at 37 degree C with SDS and proteinase K solution. Saturated solution of NaCl is added to the digest and after shaking vigorously the precipitated protein is separated by centrifugation. The DNA is precipitated by adding absolute alcohol to the supernatant containing DNA. The yield of DNA quantity is comparable to that of phenol chloroform method. Another method is Chilex extraction method. It includes 3 steps, for example, washing, boiling and centrifugation. In washing, the contaminants and PCR inhibitors like heme are removed from the blood sample. For boiling, cells are suspended in 5% of Chilex 100 which is chelating resin. Alkalinity of solution and heating it to the boiling temperature to break open the membrane destroys and denatures cell protein and release the DNA. Then the suspension is centrifuged for separating the resin and cellular debris from the supernatant containing DNA. Chilex extraction method can be non-differential and differential for extracting DNA from the stain containing sperms and female epithelial cells. This method is fast but harsh because it is having pH 10 to 11 and temperature approximately 100. It is simple but has potential for degradation of DNA during long storage. Inexpensive but traces of resin in DNA sample inhibit PCR amplification, involves less manipulation but DNA obtained is single stranded. Another method is silica based method. In this method, DNA molecules are reversibly absorbed to solid silica surfaces in presence of high concentration of chiotropic salts which disrupt the 3-dimensional structure of protein. Absorption of DNA to silica takes place due to dehydration effect of chiotropic salts which facilitate cell lysis and DNA absorption. As a result of dehydration of phosphodiester backbone of DNA, the phosphate residue interact with silica surface. This technique usually involves following steps. Number 1 is cell lysis and protein digestion step, which is carried out using proteinase K. The cell membrane is broken open and DNA is released. The next step is DNA adsorption onto silica. DNA in the cell lysate binds to silica surface in the presence of chiotropic agents, leaving the cellular material and other contaminants which inhibit PCR amplification. The adsorbed DNA is double stranded with partial denaturation. Next step is washing. Chiotropic agents and other contaminants are removed using ethanol based wash solution, while DNA is retained in the silica matrix. And next step is elution of DNA. DNA adsorbed onto silica matrix is dehydrated with aqueous low salt solution to elute DNA. The eluted DNA is double stranded. Then the technique of differential extraction is carried out for extraction of DNA from biological evidence material collected from sexual offense cases, which often contained mixture of sperm cells from male and epithelial cells from female. DNA taken out of such mixtures complicates interpretation of results. To circumvent this problem, non-sperm and sperm cell are lysed in separate steps based on differences in the cell membrane properties of sperm and other type of cells. Sperm plasma membrane contain protein cross-linked by disulfide bonds, due to which it exhibits higher mechanical strength than that of non-sperm cells. Thus, based on difference in mechanical strength of the cell membrane, first non-sperm cells are lysed in the presence of SDS and proteinase K. After separating these sperm cells from the lysed non-sperm cells by centrifugation, the sperm cells are lysed in the presence of SDS, proteinase K and DTT for extracting DNA. After extraction of DNA, the next step is quantification of DNA. After extraction of DNA, an accurate measurement of the amount of DNA and also the quality of DNA extracted is desirable. Putting the accurate quantity of DNA to PCR will yield the finest quality result in the shortest time. Adding too much or not enough DNA will result in a profile that is problematic or even difficult to interpret. This is especially important when profiling forensic sample, when it is very difficult to know the state of preservation of the biological material, and also in many cases it is difficult to estimate how much cellular material has been collected. It is less important to quantify DNA when using some reference sample, where similar amount of DNA can be expected to be extracted each time, as there are not very many variables. Even so, many laboratories still quantify the DNA from reference samples as part of their standard protocol. The quantity of DNA that can be extracted from the sample depends very much on the type of material. Each nucleated cell contains approximately 6 picogram of DNA. Liquid blood contains 5000 to 10,000 nucleated cells per milliliter. Semen contains on the average 66 million spermatozoa per milliliter. The average ejaculation produces 2.75 ml of semen. Biological sample recovered from the scene of crime are not usually in original condition and can often consist of a very small number of shed epithelial cells. Consequently, the amount of DNA that can be recovered can be extremely low and difficult to quantify. However, several commercially produced DNA profiling kits are available in the market, which are optimized to produce DNA profile from a narrow range of template DNA concentration, usually between 0.5 nanogram to 2 nanogram DNA. Too little template DNA can result in stochastic amplification, which causes unbalanced amplification of heterozygote loci and can result in allelic dropout, making heterozygotes appear as homozygotes at the affected loci. Too much template DNA can lead to the production of large Stutter peaks that complicate DNA profile interpretation. It is therefore essential that the sample are quantified before performing PCR amplification to avoid the production of such artifacts. DNA quantification is carried out using several methods. First is visualization on agar gels. In this, agar gels approximately 10 centimeter long is submerged in electrophoresis buffer and the DNA is loaded into wells that are formed in the gel by a comb. An electric current is applied across the gel and the negatively charged DNA migrates towards the anode. The agar gel forms a porous matrix and smaller DNA molecules move through the gel more quickly than the larger DNA molecules. Dyes that intercalate with the DNA double helix such as ethidium bromide can be added to the gel either before or after electrophoresis, which migrates through the gel bound to the minor groove of the double stranded DNA. DNA is visualized by placing the gel on transilluminator that emits UV light at 260 nanometer. Quantification standard are run along the unknown sample to allow the DNA concentration to be estimated. In addition to showing the presence of DNA, the size of the extracted DNA molecule can also be estimated. High molecular weight DNA can be seen as a single band, while the degraded DNA or DNA that has been seared during extraction appears as a smear. This makes comparison to the standard difficult as the DNA is spread out over a range rather than in a single band. The advantage of agar gel electrophoresis is that it is quick and easy to carry out and also gives an indication of the size of the DNA molecule that have been extracted. However, the disadvantage of this technique is that quantification is subjective. Based on relative bands intensities, it is difficult to gauge the amount of degraded DNA as there is no suitable reference standard. Total DNA detected can be a mixture of human or microbial DNA, which can lead to overestimate of DNA concentration. This method cannot be used to quantify sample extracted using Chilex, which yields single stranded DNA, and the fluorescent dyes that intercalate with the double stranded DNA do not bind to the single stranded DNA. Another method of DNA quantification is spectrophotometry. DNA absorbs light maximally at 260 nanometer. This feature is used to estimate the amount of DNA in an extract, and by measuring a range of wavelength from 220 nanometer to 300 nanometer, it is also possible to assess the amount of carbohydrate and protein that might have been co-extracted with the sample. DNA is placed in quartz cuvette and the absorbance is measured against a standard, if the DNA extract is clean, the ratio of the absorbance at 260 and 280 nanometer should be between 1.8 and 2. This technique is not species specific, therefore, any bacterial or fungal DNA that has been co-purified with the DNA of interest cannot be distinguished from human target DNA. Additionally, single stranded DNA and nucleotides cannot be distinguished from double stranded DNA by this method, which can also lead to false high quantitation reading. Another major disadvantage of this technique is that it is difficult to quantify small amount of DNA accurately using spectrophotometry. Alternatively, fluorescent dyes using pico green or ethidium bromide can also be used for DNA quantitation using spectrophotometry. These dyes intercalate with double stranded DNA and produce fluorescence, which is detected by spectrophotometer. Another method of quantification of DNA is slot blot technique. This method is based on the specific hybridization of a 40 base pair probe that binds to human alpha satellite locus. Using this technique, DNA is quantified by visual comparison of sample bands intensities with the band intensities of standards produced from known amount of DNA. The comparison can be performed manually, but this allows for subjectivity of individual interpretation, which can lead to between operator differences in quantitation, or it can be performed electronically by computationally converting the bands intensities into numerical values. This technique shows a high degree of species specificity, human or primate, and can detect as little as 150 picogram DNA in standard assay. Similar to the development of commercial DNA profiling kits, the development of commercial DNA quantitation kits has also progressed. Applied Biosystem has produced the QuantiBlot kit for human DNA quantification. Another technique of DNA quantification is using real time PCR. This methodology was first proposed by Higuchi and co-workers who, by including ethidium bromide in PCR reaction, were able to continuously monitor the production of double stranded DNA in the real time. By capturing the change in fluorescent intensity on video camera, the potential of this new development for specific DNA quantitation was realized. There are several different approaches to real time quantitation of DNA. They are all, however, based around the principle of fluorescent dye binding double stranded DNA as it accumulates during PCR process. As this technique is based on the polymerase chain reaction, DNA quantitation can be undertaken by targeting any specific region of template DNA with many systems targeting the multi-copy sequence. Several real-time PCR kits are available commercially, such as Quantifiler system from Applied Biosystem and the HY Plexure system from Promega that allow the accurate quantitation of human specific DNA and even the separate quantitation of human and human male DNA in same reaction. When generating a DNA profile, the PCR products are normally analyzed at the end point after 28 to 34 cycle. It is however possible to monitor the generation of PCR products as they are generated in real time. This was first developed using ethidium bromide. As PCR products are generated in each cycle, more ethidium bromide intercalates with the double stranded DNA molecule and fluoresces under UV light. The increase in fluorescence can be detected using suitable camera. Increasingly sensitive assays have been developed such as SYBR green or cyber green and the Talkman system. Using cyber green and PCR products are generated. The dye binds to the double stranded products and fluorescence increases. The Talkman system uses a different approach with two primers and a probe. The probe is within the region defined by the primer and is labeled on the 5' end with fluorescent molecule and on the 3' end with molecule that quenches the fluorescence. As the primer are extended by Talk polymerase, one of them meets the probe which is degraded by polymerase, releasing the probe and the quencher into solution. Efficient quenching of the fluorescent molecule only occurs when they are in close proximity on the probe molecule. As more PCR products are generated, more fluorescent molecule are released and the fluorescence from the sample increases. Real time assays are highly sensitive, human specific and are not labor intensive, and this is the technique of choice nowadays in every forensic science laboratory. Another method of DNA quantification is using DNA IQTM system, DNA quantification system marketed by Promega. This is a novel approach to quantification, and in this the isolation method is based on salting out and binding to silica. A very specific amount of silica coated bead is added to extraction and these bind to a maximum amount of DNA. Therefore, when DNA is eluted from the beads, the maximum concentration is known. It has the advantage of combining the extraction and quantification step, but has the disadvantage of not being human specific. In summary, we can say that DNA can be extracted from any type of biological sample, that is blood, semen, saliva and their stains on any surface. Organs and tissues including kidney, spleen, gonads, bones, teeth, hair root and hair shaft, nail clippings, urine, fecal matter, sweat, tear, earwax, fingerprint for touch DNA, skin, muscle tissue, dandruff and sample of plant origin, microorganism, vaginal cell on outer surface of condom, tobacco spit, cigarette bidi stub and chewing gum. Extraction methods vary according to the type and amount of biological evidence material recovered from the scene of crime and sent to the laboratory. The method chosen should yield sufficient quantity and good quality and high purity DNA. DNA of insufficient quantity and poor quality or degraded DNA may result in a partial DNA profile or failure to give any profile. Low purity DNA may cause interference during subsequent DNA testing. For example, DNA polymerase inhibitors interferes with DNA amplification. Some other considerations for selecting proper DNA extraction method include adaptability to automation, throughput, simplicity, and risk of contamination and cost effectiveness. After extracting DNA, an accurate measurement of the amount of DNA is essential, and also the quality estimation of DNA is desirable. Putting the accurate quantity of DNA to PCR will yield finest quality results in the shortest time. Adding too much or not enough DNA will result in a profile that is problematic or even difficult to interpret. Biological sample recovered from scene of crime are not usually in pristine condition and can often consist of a very small number of shed epithelial cells. Consequently, the amount of DNA that can be extracted can be extremely low and it is difficult to quantify. Thank you.